Insects communicate, in other words, exchange information by many means. Touching each other, here we refer to the tactile communication, emitting sounds, known as auditory communication, or by visual communication. However, chemical communication is probably the most extended communication mechanism among insects. In this type of communication, the insect excretes chemical substances at the environment which are detected by other insects. The chemical substances that are produced by insects for communication purposes are in general called semiochemicals. There are two types of semiochemical communication: communication between individuals of the same species, the so-called intraspecific communication, or communication between different species. Here we refer to the interspecific communication. When communication takes place between members of the same species, the chemical factor is called pheromone, and one or both species modify their behavior from this communication. When communication takes place between members of different species, the chemical factor is called allylochemical. When both species modify their behavior from this kind of communication, we are talking about pheromones. When the species that sends the signal modify its behavior, the chemical substance involved is called allomone, whereas when the species that receives the signal modify its behavior, the chemical substance involved is called chiromone. Semiochemicals are used for various purposes: to monitor an insect population, as well as to control an insect pest population using the so-called mating disruption and lure and kill techniques, or by mass trapping. But why would someone be interested to monitor an insect population? First of all, to detect if a species is present in a region. This is particularly important when we are talking about quarantine species. Moreover, it's important to estimate the population density of an insect pest species in order to know when is the optimum timing for the application of control measures, or even more, to decide which control measures to apply. Different kinds of traps are used for population monitoring purposes: odorous, visual, or complex traps, in which insects are attracted both by color and smell. Let's give now an example of how we can use population monitoring with traps in order to time schedule the control measures against the olive moth, Prey sole, in an optimum way. The olive moth is one of the most destructive insect pests in the olive production in the Mediterranean area. The olive moth has 3 generations per year: one Phylophagus generation in early spring, which feeds on leaves and lay eggs on the flowers; one Anthophagus generation in late spring or early summer, which feeds on flowers; and the third one, a Carpophagus generation, which feeds on olive fruits in autumn. There are 2 ways to estimate the size of the population of the olive moth in the olive orchards and decide when to apply insecticides: either by collecting samples of olive leaves, flowers and fruits, or by using traps. Let's say that we want to spray against the second generation when the population reaches its peak. When is the best time to spray? Before the peak, right on the peak, or after the peak? And when we say before the peak, how long before the peak? In the same way, when we say after the peak, how long after the peak? Let's have a look at the olive moth's biology. This species has 4 life stages: the egg, which is on the tree; the larva, which is on the tree, infesting leaves, flowers and olive fruits; the pupae, which is usually in the soil; and the adult, which usually flies and does not feed. Based on the insecticides that we have so far, we can spray against larvae and sometimes against eggs, but not against the other 2 life stages. Let's say that we have only one generation, the green peak that we see here at the center of this slide, which represent the maximum number of adults in the pheromone-baited traps. If we spray before this peak, this means that our population is mainly large larvae and pupae. Large larvae means that our insecticide might be ineffective, because large larvae are not so much affected by insecticides. Pupae are also not affected by insecticides, so usually the application that we may do before the peak will likely be ineffective. If we spray right on the peak, this means that the highest proportion of the population is in the adult stage, since adults fly and do not feed, they are not that much affected by insecticides. If we spray right after the peak, this means that the major proportion of the population is eggs and newly hatched larvae. Young larvae are usually more susceptible than older ones to insecticides, which means that the insecticidal application would be more effective than previously. Also for eggs, we can use formulations that have ovicidal effect, such as insect growth regulators. If we go further than that and move far beyond the peak, let's say 2 weeks after the peak, then again we have large larvae and larvae that are prior to pupation, which means that again the application might not be effective and we are already late. This also means that the next generation is coming. This is the way that we handle traps and adult captures in order to time insecticidal applications. Another technique in which pheromones are involved is mating disruption. Mating disruption is based on the use of sex pheromones in order to confuse insects and prevent them from locating each other. In this way, insect mating is disrupted and the reproductive cycle of the insect is blocked. Let's give a successful example of mating disruption with the pink bollworm, Pectinophora gossypiella. Here we see their larvae and the adults. For this species, the larva develops exclusively inside the cotton ball, so any insecticidal application would not be effective when the larvae are already there. So the larva stays invulnerable from the sprays, which at the same time kill other beneficial insects. Moreover, some small portions of insecticides enter the ball and create resistant populations through exposure to sublethal doses. At the same time, the insecticides kill other non-target organisms. This is how it goes. The female emits the pheromone signal from its pheromone glands, and in this way, the male locates the female, and mating and oviposition starts. If we put pheromone dispensers, just like the ones we see here, we block the real pheromone that is produced by the female with the artificial pheromone that is released by the dispensers. One dispenser produces pheromone which is equivalent with 200 females. In this way, the male cannot locate the female, so there is no mating, no oviposition, and the reproductive cycle is blocked. In this graph, we can see the weekly captures of male adults of Pectinophora gossypiella in pheromone traps in 2 cotton fields. In the first field, conventional insecticides were applied, whereas in the other field, mating disruption was assessed. When insecticides were applied, insect population was reduced after each spraying, however it was never eliminated. In contrast, when mating disruption was applied, there were no captures of male adults of the pink bollworm between July and August. A variation of the mating disruption technique is the autoconfusion system. In this system, the inner part of the traps is dusted with a dust with electrostatically charged particles, which carry the female sex pheromone of the insect species that we want to control. When the traps with the pheromone dust are placed in the field, the male adults are attracted by the pheromone. As males enter and move into the trap, particles of the electrostatically charged dust are attached on the insect cuticula and antennae. The dusted males now carry the female sex pheromone, making it difficult for females to locate them. Moreover, the dusted males act as dispensers of the female sex pheromone, attracting other males. In this way, when males are attracted by each other and conduct pseudo-mating, the dust particles are transmitted to other males. In this way, it becomes difficult for a female to locate and couple with a male. Even if some females manage to do so, the population growth is severely negatively affected. The lure and kill approach can also be used as an IPM strategy, which reduces the need for broad-scale applications of chemicals to crops. In this method, a semiochemical or a food attractant is used to attract target pests to a specific location, where the insect is subjected to a lethal dose of insecticide. The lure and kill technique has been successfully used against fruit flies, the codling moth, the pink bollworm, as well as against cockroaches. For mass trapping, a large number of traps must be used. Mass trapping has been successfully used against the olive fruit fly, Bactrogera olea, against the pink bollworm, as well as against major stored product insects and many other species. This is one of the oldest examples of mass trapping for the control of the cigarette beetle, Lasiodroma sericorne, in a storage facility. In this graph, we can see the reduction of the cigarette beetle population in stored tobacco over a 3-year period of continuous mass trapping. This means that continuous mass trapping will gradually eliminate the population.